Okay, so arteriogenesis refers to the enlargement of existing arterial vessels either by an increase in vessel diameter, increase in wall thickness, or a combination of the two. And we see arteriogenesis in a variety of situations. The most common and the most well recognized is in response to increases in cardiovascular disease risk factors. If we look at this image here, the center is normal, so this would be our normal vessel lumen and our normal vessel wall thickness. As we increase our exposure to various cardiovascular disease risk factors, so this would be high blood pressure, diabetes, just primary aging, those individuals, those groups, we start to see maybe some small changes in diameter, but really see changes in wall thickness. And potential mechanisms, I'm going to highlight a few here that are going to cause this type of arteriogenesis adaptation. There's going to be things like increases in reactive oxygen species, decreases in nitric oxide bioavailability, increased inflammatory processes, and increases in mean blood pressure. And hopefully you can piece together where these things would come into play. Physical inactivity, aging, going to be primary factors driving a lot of these signals that then are going to go on and result in this type of adaptation. But we also see arteriogenesis, because remember, I'm talking about vessel diameter changes and wall thickness. We can go from normal, and with exercise training, we see very little changes in the thickness of the smooth muscle that is in these would be in our larger conduit primary arterioles. We see this increase in diameter. So dilation, not just a dilation as you would think of in response to exercise. The vessel is just larger. And this is going to do to things like increases in antioxidant or decreases in oxidative stress, improved nitric oxide bioavailability, decreased inflammation, are going to be big factors. And it has on here cyclic blood pressure. Don't really worry about that too much. That's more of an experimental model. But we know with exercise training, we're going to see decreases in oxidative stress, improved nitric oxide bioavailability, decreased inflammation. And we see this. If I bring in a couple dozen of highly trained endurance athletes and compare their brachial and femoral arteries to that of those who are very untrained, you're same age, same body mass or body surface area, you're going to find the athlete is going to have a much larger vessel. And that's due to this arteriogenesis. Now, we know this is occurring both in exercise training and in response to cardiovascular disease risk factors. And this response to exercise training, while maybe it's been observed for a couple decades, it's only been really recognized here in the last 10 years. So we don't know a lot about what's driving this particular response to the exercise training. And we know even little about if you take someone who's over here and try and train them. What we do know about arteriogenesis tends to come from looking at collateral flow and changes in collateral flow. And this is largely because medicine tends to drive the research. While exercise is fascinating and very, very important, especially when you're talking rehabilitation, it hasn't always driven the research field. Coronary artery disease, however, pretty big impact on human health and medicine and all the things that go with that. So as such, it's really driven the field. So where this comes into play is if you've got two blood vessels. This can be, we tend to think of it as coronary arteries, is where a lot of this work has come from. And you've got some type of obstruction here. So an atherosclerotic plaque. As you would suspect, that is going to result in very low flow downstream from here. So you're going to have tissue that is going to have low O2 delivery and all the things associated with that. In response to this poor perfusion, we start to develop greater collateral flow. So we have these main vessels, but between them there are these smaller, less diameter, maybe less functional collateral pathways. But when you start getting this obstruction, we start getting this arteriogenesis process where we develop these collateral flows, they get larger, and we can now divert flow from this unoccluded vessel to this occluded vessel. This process is critical because you think about someone developing atherosclerotic plaque, those sorts of things. This is kind of the body's internal method in which they can take a collateral that already exists and increase its size to improve perfusion to the more anoxic tissue. So what are the signaling? We talk about this being a signaling pathways class. What are the signals that drive us going from this very small collateral to a collateral that actually is going to help function and deliver oxygenated blood? Some of the key factors, especially those in response to exercise, but even in pathology, shear stress and luminal pressure are considered to be the primary hemodynamic stimuli that prompt vessel enlargement. So it's kind of that initial signal. And if we look at a section of an arterial wall here on the right, I've got these kind of hemodynamic stimuli labeled out. So here's our blood vessel, here's our endothelial cells, and we see our intima, media, and adventitia. Shear stress is that basically, in layman's term, the friction that the blood and the red blood cells is placing on the endothelium. So the faster those red blood cells and the faster the blood flow, the greater velocity, the more shear stress is there. Luminal pressure, this would be things like just straight up blood pressure. What's the pressure inside the vessel? And we can see that here. It's the direction that these pressures are applied does in fact matter. We think they're both primary stimuli to prompt this vessel enlargement. And if you look at the responses to exercise, consistent repeated exercise, particularly resistant or endurance exercise, you're going to see changes in wall shear stress and changes in pressure. Pathologically, we get these increases in shear stress around the throat of the obstruction. So if you've got plaque buildup, and we look at that across the blood vessel. So here's proximal to our upstream shoulder. So this would be the shoulder of our obstruction, right at the obstruction, and distal to the obstruction. The flow of blood is going to have to change, and the velocity of that blood is going to be dependent where it is around this obstruction. So there's actually been some pretty cool experiments. So if we look at the percent of baseline luminal narrowing, and we look at different segments along this vessel, we've got here in red, this is low endothelial shear stress, moderate endothelial shear stress, and high endothelial shear stress. So this shear stress, this is this stress that's placed on the endothelium, and it's a stimuli that prompts vessel enlargement. We can see that proximal to the throat, there's very little difference. The endothelium is exposed to similar amounts of low, moderate, and high shear. But as we get real proximal and on top of this obstruction, we can see that we now get a greater amount of high endothelial shear stress, which is then going to drive signaling to promote vessel enlargement, whether it's vessel enlargement here or vessel enlargement on nearby collaterals. And then once that obstruction, we're past that distal downstream, we can start seeing the shear stress go away. So it's clearly a potential stimuli driving some of this adaptation in pathology, as well as in some cases exercise. So why this is important is because these endothelial cells that line our blood vessels, they can sense this changes in shear stress and convert that mechanical stimuli into a biochemical stimuli or signal. So what's sensing these changes? We've got a lot of theories, but not a lot of hard evidence. Integrins, tyrosine receptor kinases, G protein coupled receptors, ion channels, they've all been proposed. They've all got some evidence for and against them. They're kind of the top candidates. But we can't narrow down what's going on. What's really sensing at the edge of that vessel or the edge of that endothelial cell and giving us our signal. What we do know is what happens in the cell. We know that nitric oxide is released. And we see this is a response that's important during exercise. We get this increase in blood velocity, we get increased nitric oxide and causes that vessel to acutely dilate. If we kind of keep that shear stress up, we not only get some nitric oxide, but we start seeing things like the expression of intracellular adhesion molecule 1, so ICAM1, vascular cell adhesion molecule 1, VCAM1, and chemokines like tumor necrosis factor alpha, TNF alpha. And yes, while you look at the literature broadly, you can see that some of these things, can be bad. But they also are important signaling molecules in this process. And if you see some of these elevated in a patient, that might suggest some of this is taking place within the vessel. So let's go through the stages and the steps and the signaling kind of required for this arteriogenesis to take place. So there's initiation. So let's just orientate ourselves here. Here's a parent vessel that supplies an area of tissue. Over here we have a separate vessel. So these are typically referred to as parent vessels. It's going to supply its own group of or section of tissue. And we've applied some occlusion. And this is typically done within animal models where you can get full occlusion. You kind of see the massive response that goes into place. But this is going to happen in practice as well. So we've got some level of occlusion, and that's going to result in some downstream distal drop in perfusions. Low flow here. So now, because there's this low flow here, and if you had 335 or 603, you know that one of the main drivers of blood flow is a pressure difference. So we've got a high pressure over here, we've got a low pressure over here. Therefore, blood is going to be diverted through this small collateral. And it's going to go pretty quick. It's very narrow. So with this narrowing and this increase in flow, we get this increase in shear stress. So we get a drop in pressure. Over here, we get this increase in shear stress. This increased shear stress triggers an inflammatory response in endothelial cells. We start seeing basement membrane, cell recruitment through expression of adhesion molecules and cytokines. So we just talked about ICAM1, VCAM1, TNF alpha, all being upregulated with shear stress. This is where it's happening. We get this increase in shear stress. That activates our endothelial cells. We get increases in chemokines and adhesion molecules. So this initiation, this is the initial signal to tell the cell to start enlarging, or tell the vessel to start enlarging. We're getting the release of these VCAM and ICAM and TNF alpha molecules. And they actually serve as both paracrine and autocrine signaling. We talked about that early in the semester here. So paracrine and autocrine signaling through cytokines and growth factors leads to smooth muscle differentiation from a contractile phenotype, indicated in the image as red, to a synthetic phenotype, which is gray. And that starts to build this what's called translational matrix. I'm not going to highlight that. Just to highlight here that, these molecules that are being produced, they're called through paracrine and autocrine signaling, changing these smooth muscle cell function from being more contractile, what we think a smooth muscle cell is going to be, to more of a synthetic, where it can allow for changes to take place. It can allow for the vessel to enlarge. So we see here in our kind of second step, basement membrane recruitment, we see adhesion and recruitment of our basement membrane cells. We see breakdown. And the key thing here is we get this switch to of smooth muscle cells. So that's the SMC. Smooth muscle cells switch to a more synthetic phenotype. We're all still signaling here. The next step is the growth step. We start getting the growth phase. So we're getting luminal expansion, vessel lengthening, and we get a tortuous pattern. So you'll notice that in the image here, it's not this nice straight line that initially was. It's very tortuous. And if you actually look at images, whether it's an ultrasound or nuclear imaging, you can look at collaterals and see that they are very, very tortuous. They're not nice and smooth and straight like a brachial artery would be or any downstream arterioles. And these are characteristic of growing collateral vessels. As they increase, as you'd expect, we start seeing a decrease in resistance. We get a restoration of flow down here. And shear stress begins to normalize. It's widened out. We've removed that stimulus, and it tells the vessel to stop growing. So we get some smooth muscle cell and endothelial cell growth and outward remodeling. That gives us our luminal expansion, as we would want. We get, with a larger vessel, we restore perfusion to this tissue over here. That allows the shear stress to become normalized and our inflammation to go away. So we've had our initial signal of the increase in shear stress that activates the endothelial cells and causes it to basically increase inflammatory factors like TNF-alpha, ICAM, and VCAM1. Those signal themselves, basically the smooth muscles, and tell them to change their phenotype. That allows for luminal expansion, so things kind of work their way out. We get, therefore, this larger vessel. We get normalized in shear stress, normalized in flow, and the inflammation goes away. And we've restored flow downstream. This is happening in not just one collateral vessel, but if there's multiple collateral vessels involved, they're all going to start going through this process. But they all don't complete the process. So there is kind of this maturation phase, as it's called, that the vessels that develop with the least resistance and therefore supply the most blood flow, they go ahead and mature into this dominant collateral that's there to stay and is going to supply blood to the area. Other smaller collaterals that maybe didn't develop quite as fast, the stimulus wasn't there, whatever it may be, if they can't really sustain that hemodynamic stimulus, they'll kind of regress back to the normal small collateral flow that's there. So the vessels with the least resistance supply the most blood flow and therefore mature into the dominant collaterals. And we can see that either or situation here in our image. On the left, we've got high flow, good diameter, very low resistance, and this is going to mature into our dominant collateral. And our smooth muscle cells, they'll return back to their contractile phenotype so that they can modulate vascular resistance. Or, so the left half would be in one part of the circulation, on the right it could be somewhere else. If there's Really not much adaptation, not much increase in vessel diameter. It will eventually go back to its normal state where it's very small and not much flow. So that is kind of the key steps with arteriogenesis. It's the enlargement of an already existing vessel. What we're going to spend most of today's class dealing with is angiogenesis, which is the formation of new capillaries via extension or remodeling from existing capillaries. If you look at the literature in regards to angiogenesis, especially in today's world, you're going to see mountains of data when it comes to tumor angiogenesis. And that makes sense. It's a pretty important topic. If you look a little bit later, earlier data, so 10, 20 years ago, you'll see a little bit of the initial stuff was on wound healing, and there's still current wound healing related literature out there. But most is in regards to pathology and tumor growth. The few physiological circumstances where we can prompt angiogenesis is ovarian cycling, placental development, exercise, and wound healing. We're going to talk, and I'm going to show a little bit of data here in response to wound healing and exercise, because I think they really relate to a lot of the interests that we have here in the room. And this is a pretty quick response. So let's just look at two images here. So this is a control animal. We can look at, here they've got highlighted the capillary network. They then induced a wound stimulus and look 60 hours after wounding. And you can see within 60 hours the dramatic increase in capillary density. So it's a very important wound healing response, this angiogenesis. So if you're interested in wound healing, this is a lecture for you. We also know that capillarity in active muscles is increased by training, particularly endurance type training. We'll see where that kind of comes into play here in a couple slides. Why is that important? Well, if we increase the capillary density, that increases our potential surface diffusion, O2 diffusion. If you're one of those that believe that the diffusion path is important in humans, then that's going to help that, and it's going to increase the length of time for diffusive exchange. You get a longer capillary, you're going to therefore increase the potential for O2 diffusion. And if we look at some actual data, we can see this does take place. So this is change in capillary to fiber ratio. We look at, here's our cross-sectional of a muscle, here's each individual muscle fiber, and the little dots are capillaries. So we can report this as a percent or some sort of a ratio. And if we look at conditioning programs, so this is in animals, the velocity, so here's low velocities, 2.5 or 5 minutes, 15, 25 minutes, or a high velocity, 5, 15, and 25 minutes. We can see this pattern. If you exercise longer, you're going to get an increase in capillary fiber ratio, and if you increase at a greater velocity, you're going to get an increase in capillary to fiber ratio. And if we actually look at mean number of capillaries around a given muscle fiber as a function of VO2 max, this is a pretty cool relationship. Get that high VO2 max up here of 70 and 80 mils per kg per minute. We're talking about 6 to 7 capillaries per muscle fiber. And then down here, while this is by no means low, 40 and 50, we're down here to 3 or 4. So this is a response that we see in exercise training and in wound healing. There's two types of angiogenesis. One that we know very little about and one we know quite a bit about. Intussusception angiogenesis refers to the process by which a single capillary just splits into two. I'm going to come down to this image first, and then I'll come back up and highlight a few things. So with this type of angiogenesis, we've got a capillary here. Here's our capillary lumen. We can look at the cross-sectional. Here's our endothelial cells. We've got basement membrane around it. And basically we get this pinching effect. It pinches, and eventually we end up with two capillaries. That's really all the process involves. So we get this capillary splits into two capillaries from within by the formation of a pillar-like structure or longitudinal divide on the luminal side of the capillary. Basically forms two tubes through which a blood vessel can pass, or blood can pass. Most of the literature to date has really focused on demonstrating this actually occurs. And there's pretty good evidence that it does. We know little to nothing about how it occurs. So it's occurring, but we don't know how. What we do know occurs is sprouting angiogenesis. It's been well documented, and we have really good understanding of what's causing this. And if you actually look, if you've looked at some of the film from Dr. Mush and Dr. Poole's lab, where they actually count red blood cells going through the capillary network, you can see that it's not just a whole bunch of straight tubes, it's this intersecting highway. And we can hopefully think about visualizing the sprouting that occurred, this connection, and how we can increase the connection between these capillaries. And this often comes through this sprouting angiogenesis. So it's the process in which an activated endothelial cell branches out from an existing capillary, extends through the surrounding matrix or tissue, and forms this cord-like structure. And it usually connects to some other type of capillary venule to become very functional. So here's our red blood cell, here's our endothelial cells and our capillary lumen, and we start getting this little bit of sprout. So we get this projecting out, and we eventually get this sprout. And if you actually look, so this is one of the cooler images I think we have in this class, is here's a cross-section of a capillary. And then if we apply, what we're going to talk about is VEGF here in a second, to this capillary, you can start to see the sprouting occur. And these are these little sprouts extending out to expand the capillary network. So these newly formed tubes must enter the capillary and basically bind with another capillary venule to become very functional. So the signals for this, traditionally hypoxia, endothelial shear stress, or damage caused by some form of tissue injury, were considered the primary stimuli to stimulate angiogenesis. So if you look at the 10 to 20 to 30-year-old literature, those are the primary drivers. And there's still common literature today that uses things like hypoxia and damage to initiate angiogenesis and study it. For a long time, yeah.
Is hypoxia in this case like something that's supposed to be pathological, or just this state of being like an exercise state so much that you're not getting enough oxygen and you're like high intensity or long duration exercise?
That's what was initially thought. But in exercise, tissue never becomes anoxic. If you look at the PO2s, you're not at the capillary pressure, capillary PO2, it hangs out there around 15 to 20 millimeters mercury. If you look at the cell culture studies where they say it's hypoxia, it is way lower than that. So the hypoxia does not appear to be an issue and a stimulus driving these adaptations in response to exercise. You know, the non-physiologist would say that it is, but it's because they don't know the literature. So it's kind of one of those. So you'll read these papers from biochem labs, and they're right. They'll take a tissue sample and they'll drive it really low hypoxic gas, hypoxic environment. And they stimulate and they go, oh, this is probably what's causing exercise. But you guys, because of the classes that we teach here, and this is where integration of this knowledge comes into play. Is if you take Pool 796 or some of those other classes, you'll get into what are the PO2s in these tissues, and you'll realize, oh wow, we don't ever really become hypoxic, even at high altitude. The tissue is not getting to those states. So yeah.
So then, what's like a situation where that tissue would become hypoxic?
So the situation would be very pathological. So in a situation where you've got an atherosclerotic plaque, and the problem is this is going to take some time, and that tissue is usually going to die before then, like with an MI. So hypoxia is one of those that it's kind of a laboratory thing, not one of those things that we think is really driving it. What we do think is driving it in response to exercise, there's really three things. The first is endothelial shear stress. So during exercise, there's this angiogenic center. I'll show you here in a slide or so. So this angiogenic center releases these pro-angiogenic molecules. And we know that endothelial shear stress, so this is again that friction within a blood vessel, due to increases in blood flow and blood velocity, are going to increase some of these pro-angiogenic molecules. So here is static versus high shear stress condition, and this is vascular endothelial growth factor A. And you'll become very familiar with that here in a couple pages. It is one of these pro-angiogenic molecules. That's a pretty profound increase with shear stress. So we think that, and we know that shear stress is going to go up in response to exercise. There's also some evidence for neuronal nitric oxide. So this is generated by muscle activity. They've done some work here, not in my lab, but in a couple of the animal labs, looking at neuronal nitric oxide. And if we look at that neuronal nitric oxide and with stimulation, and then we stimulate that same muscle, but we block the neuronal nitric oxide and look at vascular endothelial growth factor levels, we can see under stimulation, VEGF goes up. So again, VEGF is going to be one of those signaling molecules. We start seeing this increase. If we block neuronal nitric oxide, it's very blunted. Eventually it works its way back up, but it's blunted. So we think neuronal nitric oxide may be playing a key role. The third, you guys are already writing down, is the transcriptional coactivator peroxisome proliferator activated receptor alpha coactivator, PGC1 alpha. Hopefully you've remembered that PGC1 alpha is in skeletal muscle, that it is upregulated with endurance type activity. It's exercise intensity independent. And some of the coolest evidence for PGC1 alpha is if we take mice that lack PGC1 alpha in their skeletal muscle and we train them, they fail to increase capillary density in response to that training. So if we wipe out PGC1 alpha, we don't see any increases in capillary density. And that's this data here. So this is vascular endothelial growth factor protein levels. The WT means wild type. They're just a normal mouse. The knockout means that we've genetically removed PGC1 alpha. And you can see that control and trained in the quadricep muscle, the wild type, see this big increase in VEGF. And the knockout, very little, not even a significant increase. If you remember back to our lecture where we talked about the interaction between resistance and endurance type training, we looked at animals that were given this AI-CAR, which increased AMPK levels and therefore increased PGC1 alpha, we can then start to see things come into play that clearly PGC1 alpha is an important factor. And if we look at kind of the pathway, the potential pathway, and this over here is what's really important. You know, obviously this is a potential thing. So here's our skeletal muscle. And I don't have it written up here, but go and put endurance exercise right up here at the top. Well, we know endurance exercise is going to alter the ATP to ADP ratio. We learned that in the last module. That's going to increase AMPK. And we know from the last module that's going to increase PGC1 alpha, which normally we'd put an arrow off here and that would go and increase mitochondrial biogenesis. We'd get these great skeletal muscle adaptations. But in this lecture, this PGC1 alpha is also increasing vascular endothelial growth factor expression, which we'll learn here in a second, increases the proliferation of endothelial cells and contributes to angiogenesis and capillary density. So there's really several things that could come into play in response to exercise that are driving this increase in vascular endothelial growth factor. You know, damaged cells themselves, the reason I'm not talking about the damage side is you damage the cells, they're going to increase this VEGF as well. That's pretty well established. So let's talk about this VEGF. The main proangiogenic molecule is vascular endothelial growth factor type A. It doesn't function on its own, it likes a partner, so it always dimerizes with another VEGF A molecule. So on the left here, this is an individual VEGF molecule. This is the dimerization state. The dimer, there's just two of them. You've got to have the two in order for it to interact with its receptor. All right, in point 15, there's a slight error here. So this is VEGF-R1. So VEGF type A binds to two primary receptors on endothelial cells. The VEGF-R1, so VEGF-R1, and VEGF-R2, which is just vascular endothelial growth factor receptor 1 and receptor 2. While it can bind to both, it's pretty well accepted that VEGF-R2, so the receptor 2, elicits the complete angiogenesis response. And there are a variety of these vascular endothelial growth factor type receptors, and that's what I've got shown down here in this image. We've got VEGF-R1, R2, R3, so on and so forth. This one here, so here's our VEGF-A. There's different subtypes. You don't really need to highlight this. It's just so you can kind of see where I'm following here. These bind to this receptor. So here's our VEGF-2 receptor. And that's going to be involved in proliferation and migration and endothelial cell survival. All right, so now that we've established that kind of our main signaling molecule is this growth factor A, it's binding to this receptor type. Let's go through the actual process now that drives this increase in capillary density in response to damage and exercise. So we've got here on the top is a capillary, at the bottom is a separate capillary, and then we have tissue in between. The angiogenic center is just whatever that tissue, whether it's been damaged, maybe it's PGC1 alpha, whatever it may be, is causing the release of vascular endothelial growth factor A. So we've got some tissue here, and it's releasing VEGF. And it's going to do that, as we've kind of already mentioned. So if you look at a lot of the cell culture literature, so hypoxic environment, site of a wound in which immune cells like macrophages are going to increase or release VEGF. Some endothelial cells can release it, but more importantly, within response to exercise, skeletal muscle. So we've got some angiogenic site. At this point, any of these factors could release VEGF. And as we would expect, it binds to its receptor. And all of these endothelial cells are going to have this VEGFR2 receptor. It's just the cell that has the most receptors and gets the most VEGF is the one that gets to play the game. It becomes what's known as the tip cell and becomes very, very specialized for this angiogenic process. So we can see here, some of these molecules are going to go over here and some are over here. These tip cells, as I've illustrated here, have a couple different receptors and are going to get the greatest signal because they get the highest dose of VEGF. And we've got a tip cell on both capillaries. And this starts the signaling to tell that cell to become extremely specialized. As you can see here in the image, it looks very, very different. So once it gets a high enough of VEGF, it gets these extruding protrusions that begin to migrate towards the source of the growth factor. So it's migrating towards the angiogenic center. So this angiogenic center is constantly releasing the VEGF molecule. These little protrusions are called filopodia. They're covered in these receptors, and they need to be. They need to know where that signal's coming from. So it's directing that tip cell towards the portion of tissue that is releasing the signal, whether it's damaged, skeletal muscle that's releasing the PGC1 alpha, whatever it may be. So it's directing that tip cell towards the source. Now, what we don't want is just rampant number of tip cells. We don't want all these endothelial cells thinking they all get to play this game and turn themselves into a new capillary. So there's got to be some sort of signaling to prevent that from taking place. So that's kind of point 9 in this image here. The tip cells also express something called delta-like ligand 4, DLL4, that binds with neighboring cells. So it binds with the neighboring endothelial cells' Notch receptors. So if we look here in our image, here's our filopodia. They're releasing this DLL4. Neighboring endothelial cells have these little Notch receptors, and it forces these neighboring cells to stop expressing VEGFR2. Because if they keep getting that signal, they're going to start developing filopodia, and this whole process gets out of whack very, very quickly. We learned several weeks ago about the different ways in which we can remove a receptor. It can be internalized, it can be just completely destroyed, it can be just inactivated. But these DLL4s bind to these Notch receptors and cause the neighboring cells to stop expressing. One of the theories with tumor growth and tumor capillarization and some of those things has been that some tumor cells may not have these Notch receptors, or at least not as many, and therefore we get this kind of rampant capillarization of these tumor cells. So that's the key thing here. We've got to have these Notch cells and these DLL4 molecules. The filopodia also need to release proteolytic enzymes, because it's not just open air. They're working their way through tissue. So if we're talking skeletal muscle here, it's got to work its way through that skeletal muscle, which means there's got to be some breakdown of the tissue. So these proteolytic enzymes are going to break down the basement membrane and other components of the extracellular matrix that allows that tip cell to migrate towards that angiogenic center. So that's all just starting out. That's just the tip cells and their nice little philopodia. As this progresses, that tip cell begins to further migrate towards that angiogenic center, and the neighboring cells don't necessarily move with it. They proliferate, so they multiply, and we get new endothelial cells. These new endothelial cells are called stalk cells. So if we look down here at our image, I'll come back up in a second. So here's our capillary, our angiogenic center, our other capillary. The new stalk cells are developed. Our philopodia are working our way out. And at this point, we don't have a new capillary lumen, but what we have is called a sprout. We're getting this building of endothelial cells, the philopodia are reaching out, and we're getting this sprout. And we just keep adding stalk cells and reaching out until the tip gets close enough to the angiogenic center to kind of stop the process. So it's happening on both sides. So that's point 22. Different parent capillary, so we have multiple stalks progressing and migrating towards this angiogenic center. We start to develop vacuoles, so that's going to be the open space along the stalk. These eventually combine and give us a continuous tube within, that should be stalk, and then we end up with our continuous tube. Once we have the two tip cells meet their stalks, they combine, resulting in a new capillary that connects the two parent capillaries. Because now we've gone through this entire process, the angiogenic center is now no longer producing the growth factor, and the whole process can shut down. So that's what VEGF does. It helps initiate and drive this entire response. And there's a lot of things going on here. We've got different signaling, we've got proliferation of different cells, we've got differentiation of endothelial cells. So this VEGF molecule is driving this response here. It wouldn't be a signaling pathway class unless we actually talk about what's happening after that VEGF binds. Yeah. How far is it going to diffuse? Yeah. So I don't know if they actually know that. I think it's fairly localized, because if you're going to actually generate this response, if you think back to the image, and part of it's going to depend on how big your angiogenic center is. But if you're talking about damaged tissue, so you've got a pretty widespread damage, that's going to be a lot of VEGF, and you're going to get a lot. Same with skeletal muscle. You're going to have a lot of adjacent skeletal muscles producing these things like PGC1 alpha, and therefore. So I don't know if they actually know how much the inhibitory, what's the distance it can drive. From what I've seen of trained and untrained micrographs of skeletal muscle and capillary density, is that they're not on top of each other. They're fairly close. So, I'm guessing within a few fibers, but I don't honestly know. That's a great question. Other questions? Okay, so the VEGF2 receptor, we know it's composed of various domains. The main thing here, it's got a tyrosine kinase domain. So here's our cell membrane. extracellular and intracellular. Extracellular, here's our dimer of our VEGF dimer, binds to the receptor, and we've got these tyrosine kinase domains on the inside. So with binding, we know that we should get activation, autophosphorylation of the tyrosine kinase, and that's going to upregulate, activate PI3K. I'll let you guys write here a little bit, and then I'll talk about it. So we get, let's just highlight the key steps here. The VEGF dimer binds. We get autophosphorylation of the tyrosine kinase. And we get activated PI3 kinases. That then takes us from PIP2 to PIP3. So let's step here. It's going to activate AKT, or protein kinase B. Again, goes back to, and this process you should know. We just talked about this within skeletal muscle. The only difference is now we're in endothelial cells, so we get AKT, and that activates mTOR1. I'm skipping the TSC and Reb. I've already tested you over that, so I don't need to. So we've got these basic steps: VEGF, tyrosine kinase, PI3K, PIP3, AKT, and mTOR. We know mTOR can integrate a lot of this information and upregulate a variety of transcriptional translational factors. Because there's so much going on in these endothelial cells, we've got to have multiple pathways that this VEGF is going to turn on, not just this mTOR pathway. So we also get activation of phospholipase C. We talked about phospholipase C early on in the semester when we were talking about inter and intracellular signaling. Phospholipase C, we know, is going to impact the DAG molecule and the IP3 molecule. And like always, I'll give you both the name and the abbreviation. So diglycerol, the DAG molecule, and the IP3 molecule. IP3 results in calcium release from the endoplasmic reticulum. That, with the DAG molecule, activates protein kinase C and promotes our RAS molecule. And this will make some sense here in a second when I show you the image, because this is all stuff you've seen before. It's now actually applying it to a process that we care about, like wound healing or capillarization with endurance training. We know that calcium interacts with DAG and activates protein kinase C. Hopefully you remember a little bit that the RAS molecule, if it becomes activated, results in a particular pathway. So let's just talk about this here. We've got our VEGF molecule. Let's see if we can highlight that. Tyrosine kinase activation. Phospholipase C, we already did this. That's from the previous page. I'm just kind of integrating it all together. So phospholipase C. Here's our IP3 and our DAG molecule. The IP3 gives us a little calcium. Calcium and the DAG molecule give us protein kinase C. Oh, sorry about that. That gives us RAS. And we know from early on in the semester, again from the inter and intracellular signaling lecture, that the RAS molecule stimulates the MAP kinase pathway. So we're talking about the MAP kinase, and so on. And one of those that was pretty prominent was the RAF, MEK, ERK pathway. And those then turn on some transcriptional factors like MyC and CFOS and CJune. I have you write those things down, but I'm not going to test you over those. Again, getting a little overzealous at times. So the activated RAS molecule triggers phosphorylation cascade of the MAP kinases, mitogen-activated protein kinases. And the importance of that is the MAP kinase pathway allows that ERK, that final step, then actually goes into the nucleus and can have an impact on transcription factors. So it can change that conversion of DNA information into mRNA information, and then that's going to have an impact on the cell. The last pathway that's going to go with VEGF deals with calcium. So that calcium that was released interacts with calmodulin and gives us the calcium-calmodulin complex. Again, pathways that we've already introduced you to, now we're putting them into practice. So calcium interacts with calmodulin. We get our calcium-calmodulin complex, which activates calciurin. And the calciurin works on the transcription factor NFAT, or nuclear factor of activated T cells. This is important because this particular pathway we know is going to be involved in cardiac muscle, skeletal muscle, nervous and vascular tissue. And therefore is likely going to play a pretty key role into the exercise adaptations. Not just in skeletal muscle, but we kind of like good capillary density in cardiac muscle as well. So this is an important pathway. So this highlighted here, we've got our VEGF, our tyrosine kinase, phospholipase C, IP3, calcium, and in addition to going through this RAS pathway, we get calcium, calmodulin, calciurin, and nuclear factor. And these are all going to alter gene expression, so they're going to be involved in cell, particularly endothelial cell proliferation, survival, vasopermeability, and overall angiogenesis to increase capillary density. That should be it for this lecture. Questions?